Hello students, today we are going to discuss about restriction enzyme. It is a protein that recognizes a specific short nucleotide sequence and cuts the DNA only at that specific site, which is known as restriction site or target sequence. More than 400 restriction enzymes have been isolated from the bacteria that manufacture them. What are restriction enzymes? Molecular scissors that cut double-stranded DNA molecule at specific point. Found naturally in a wide variety of prokaryotes. And it is an important tool for manipulating DNA. So this restriction enzyme is also known as molecular scissors. In other words, restriction enzymes are also called as restriction endonuclease. It is an enzyme that cuts double-stranded or single-stranded DNA at specific recognition nucleotide sequence known as restriction site. So this is the snap where we are going to discuss briefly. Okay, so here you can see the restriction enzymes. Okay, so the orange color box which denotes the ECOR R1, that is the restriction enzymes. So this we are going to discuss. Restriction enzymes or molecular scissors are used for cutting DNA. So what are the steps to follow? Two enzymes from E. coli that were responsible for restricting the growth of bacteriophage were isolated in 1963. One of them added methyl group to DNA and other cut DNA into segment. So the later it was called restriction endonuclease. So E. coli refers here Escherichia coli. Okay. The first restriction endonuclease in 2 was isolated by Smith, Wilcox and Kelly in 1968. They found that it always cut DNA molecule at a particular point by a specific sequence of 6 base pairs known as recognition sequence. So the base pairs is like this: adenine to thymine, cytosine to guanine, guanine to cytosine and thymine to adenine. So these are the nucleotides which were based on the sequence. So on the specific sequence of 6 base pair, so that is known as recognition sequence. Besides N2, more than 900 restrict enzymes have been isolated now, okay, at present, from over 230 strains of bacteria, each of which recognize different recognized sequence. Naming of restriction enzyme. So how we can name this restriction enzyme? For example, if it is like ECO R1, so how it will denotes. So the first letter is derived from the genus name. Okay, so ECO, that is E, is a genus name, whereas Escherichia. And the next two letters from the species name of the prokaryotic cell from which enzymes are extracted. So ECO, so that is COS coli. So the E is the genus name and CO is species name. So whereas ECO R1 comes from Escherichia coli RY13. So these are the strain and these are the enzyme which is extracted from the bacteria called E. coli. The Roman number after name show the order in which the enzyme were isolated from the bacteria strain. So the Roman number indicates the order of the bacteria. Okay, the order of isolation of enzymes from the bacteria. So this way we have to name the restriction enzymes. Restriction enzymes belong to a class of enzyme called nuclease. So please make a note. Restriction enzymes belong to a class of enzymes called nuclease. Now this nuclease are of two types. One is exonuclease and other one is endonuclease. Whereas exonuclease, they remove nucleotide from the end. Whereas endonuclease, they cut at the specific position within the DNA. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in DNA. So second point, the palindrome in DNA is a sequence of base pair that reads same on the two strands when orientation of reading is kept same. For example, the following sequence reads the same on the two strands in 5' to 3'. So the direction as well as 3' to 5' direction. So this will be like palindrome sequence. So now here you can see the example. Okay, so the 5' guanine, adenine, thymine and cytosine, and ends with the 3'. And next here you can see the 3' CTTAG. So this will be like called palindromic sequence. Here we studied, right, palindrome in DNA. So this is the palindrome in DNA. For the example of palindrome, the other word we can refer as Malayalam. So Malayalam is the term, is the word where you can read from the beginning or where you can read from the ending. So the term, the words are equal. So that is palindrome. So this is the palindrome sequence. Mechanism, action of restriction enzymes. So here, these are the steps in the formation of recombinant DNA by the action of restriction endonuclease enzyme EcoR1. So now I'm just going to explain by the step. Later we will discuss with this image. Restriction enzymes cut the strand of DNA a little away from the center of the palindrome site, but between the same two bases on the opposite strand. So here you can see the palindrome site. The enzymes cuts both the DNA strand at the same site. Okay, where in EcoR, that is, okay, where the restriction enzymes, EcoR1 cuts the DNA between bases G and A only when the sequence GAATTC is present in the DNA. So this leaves single strand portion at the end. They are overhanging stretches called sticky ends on each strand as given in above figure. So these are named so because they form hydrogen bond with the complementary cut counterparts. So here you can see the sticky end. Okay, so once the EcoR1, the restriction enzyme we've used, where it acts as a molecular scissor and cuts the DNA molecule. So where you can see the sticky end. The stickiness of the end facilitates the action of the enzyme DNA ligase. So this sticky end where it helps to stick it, that is the DNA ligase is another enzyme where it acts as a molecular glue to fix it. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different source or genomes. So now these sticky ends are complementary to each other when cut by same restriction enzyme, therefore can be joined together end to end using DNA ligase. So in this snap you can see the step in the formation of recombinant DNA by the action of restriction endonuclease enzyme EcoR1. So here you can see the vector DNA, on the right hand side you can see the foreign DNA. So in the vector DNA, the enzymes cuts both DNA strand at the same site. Whereas in EcoR1 cuts the DNA between the bases G and A only when the sequence GAATTC is present in the DNA, that is in a palindrome condition. So next step is EcoR1, that is the restriction endonuclease is an act and where it helps to cleave the DNA. When the DNA gets cleaved, so there will be like sticky end to occupy. So that sticky end here is refers to with the help of the DNA ligase, it helps to reglue it, that is rejoin it. Okay, that is it can help to molecular glue. It can able to fix it. Once the sticky end is fixed it, so there will be the combination of two DNA. So that process we will call as recombinant DNA. So these are the step in formation of recombinant DNA by the action of restriction endonuclease enzymes. Thank you, students.